This is a production of Cornell University.
I haven't actually heard anyone read the title of my thesis out loud, but I do regret making it quite that long because when I printed my actual dissertation, most of that just got cut off. So, you know, for those of you graduating soon. So thanks everyone for being here. Thanks for providing me with this opportunity to share my work with the department. As Paul mentioned, I was an undergrad here not too long ago, and then just recently moved back from Berkeley. So for this seminar, I'll be discussing the prolific bacterial plant colonizer, Pseudomonas syringae. I'll start by describing this particular study system, and then I'll share my work adapting randomly barcoded TnSeq to screen for traits that Pseudomonas syringae requires to colonize the leaf surface and the leaf apoplast. Finally, I'll share some unpublished data expanding this work to alternate host plants and some of the other directions that I'm moving with this technology to better understand plant bacterial interactions. Okay, so before jumping into the methodology, I want to take a moment to give some background to the pathosystem that I'll be focusing on. So Pseudomonas syringae is a really common bacterial plant pathogen. It's a widely used model, partially owing to the early availability of genome sequence for multiple strands. It's capable of infecting many different plants, but a single strain will typically only infect one or a few. Much of the specificity has been linked to the dozens of effector proteins secreted by the type 3 secretion system, which help mediate the close relationship that this pathogen has with living plant cells. So here I'm showing different strains infecting tomato and bean. And I want to mention that even within a single plant like bean, you can have multiple strains infecting the same host. So there's a lot of diversity within this single bacterial species. Importantly, the species is found globally throughout the water cycle. So in addition to agricultural waters, it has been isolated from glacial snowpack, rivers, and other water sources that are far from crops. One physiological property that's thought to contribute to the spread is ice nucleation. This is a property where an individual or a few cells produce a surface protein that's actually capable of ordering water molecules to promote them freezing at a slightly warmer temperature than that water would freeze otherwise. This is thought to contribute to cloud formation and other movement of water with associated cells on a global scale. On a more local scale, this property can lead to increased frost damage. On a more positive note, this trait has also been adapted commercially to use in making artificial snow. And this does not require the use of living cells. I have a quick example of what ice nucleation actually looks like in real time, if it works. So some strains are more active than others. It's really dependent on the quantity of ice nucleation active or INA surface protein. In the past, this property has been used as a reporter for gene expression, but we, in our lab at this point, we primarily used it as a demo for science outreach events. Gets people's attention. So thinking about the many environments where Pseudomonas syringae can be found, it can colonize different plant habitats as well as be found in chance water sources. These represent various biotic and abiotic challenges to survival. Even a single plant or a single leaf represents multiple potential habitats at the scale of an individual cell. So for a typical infection cycle, you have cells land on the leaf surface, often through rain splash or movement from plant debris. Cells can reach relatively high population sizes on the leaf surface. This is their epiphytic growth stage. Some strains are a lot better at this than others. And eventually, they're capable of moving through, typically stomata, to colonize the leaf apoplastic space, so actually within the leaf itself, before reaching, and they can reach at that point, relatively high population sizes and cause visible disease symptoms. So these strains maintain many different compatibility traits that are necessary for host colonization at these different steps. And one of our broad overarching questions is, what genes are needed when? We know that growth on the leaf surface can expose cells to light, moisture, and temperature fluctuations, as well as nutrient limitation, among other things. And the apoplast presents the additional challenges due to the specific and often induced plant defense response. So Pseudomonas syringae is a very diverse species complex. A recent publication from David Gutman's group pulled genome sequence from around 500 different strains. So with this relative wealth of sequence data, Pseudomonas syringae is a really great system for comparative genomics. I thought this was a particularly nice example from a recent review of reconstructing the evolution of necessary virulence traits using genomic comparisons. So this is a proposed evolution from a non-pathogenic common ancestor with a Pseudomonas fluorescens-like ancestor to the gain of specialized virulence traits. These are ranging from some ancestral traits like the production of the polysaccharide alginate, the ability to cause water soaking in plants, which are included among other traits thought to allow broad environmental colonization and survival. And then more recently, variation in type 3 effector proteins, phytotoxin biosynthesis, ice nucleation, and other traits are thought to more specifically be adapted for plant host colonization. But for my focus, moving from comparisons between strands, I want to focus on a single strand. So in this case, B728A, which was one of the first 3 syringae strains to be sequenced. Surprisingly, a very large proportion of the genome has a completely unknown function. And this is a model strand that's been studied for many years. For other organisms, this number can be significantly higher. And Laura Huberman touched on this last week for many fungal species as well. Often homology to known genes is not sufficient to assign protein function. And so we need tools to more quickly screen more genes within diverse genomes for functionality. So how can researchers identify what genes out of many are important in a given condition? One strategy is to use gene expression and gene induction as an indicator of presumably important genes due to their active transcription in one or more conditions. So with this example study in B728A, they identified functional categories of important genes for growth on the leaf surface and in the apoplast. So for example, flagellar synthesis and motility genes were relatively highly expressed on the leaf surface compared to genes such as those involved in compatible solute synthesis, which were generally involved, generally highly expressed in the apoplast, but also relatively highly expressed on the surface as well. Typically, when you've identified genes that are potentially of interest in a method like this, you'll want to test this. Often what people do is make knockout mutants and test these for growth phenotypes one by one. And this was really my motivation for setting up TnSeq in this system. Early in my PhD, I constructed and I screened many single gene knockout mutants where these genes were either highly up or down regulated in planta. But these mutants often had no growth phenotype. And so this led to my interest in TnSeq to functionally examine gene contributions to fitness in a random and generally unbiased way. So TnSeq, or Transposon insertion site sequencing, is essentially a massive competition assay. You can make inferences about relative mutant fitness within a heterogeneous population by comparing population changes over time, typically after exposure to some sort of stress condition or selection. In some cases, competition assays can actually be more sensitive to small differences in fitness than doing parallel growth measurements. The goal here is just to identify important genes in various conditions by actually measuring their contributions to fitness and growth. And I'll just remind you that these are all relative measurements. And so using this simplified figure, where the vertical lines here are representing essentially the counts of many independent transposon insertions throughout these 3 non-essential genes. And you can infer gene contributions to fitness by changes in the relative abundance over time. So for genes such as this red one that are important for growth, the final population will be depleted in insertions in that gene. You can use this to calculate a ratio of gene fitness. And the values are going to range from large negative values, indicating that that gene was highly important under that condition, to positive values, which indicate that the gene was actually detrimental under that given condition. And this is a really powerful method to estimate gene contributions to fitness for most or all non-essential genes. The challenge with this method really comes down to the difficulty in identifying and quantifying the transposon insertion sites. And so this is where the recently developed randomly barcoded or RB-TnSeq comes in. This is just one strategy to make this process easier. All that's really changed is how these counts are generated. So for each insertion, a random 20 nucleotide barcode is carried along with the transposon into the genome. And so to actually measure these insertion counts, rather than a traditional library prep, which involves random fragmentation of the genomic DNA, adapter ligation, PCR enrichment, sequencing, and insert mapping, you simply need a single PCR reaction to amplify all the barcoded regions within universal primer sites. And this is done for all mutants in the population simultaneously. These PCR primers also include Illumina sequencing adapters and indices, and so these samples can be multiplexed. So TnSeq has historically been a really powerful approach to screen for gene function, but it requires laborious sample preparation and analysis, as I've mentioned. It also requires longer sequencing reads because you're reading from the transposon insertion into the surrounding genomic DNA to determine where that insertion actually is. Here, for this method, TnSeq is used just once to map the barcode insertions to their location in the genome. And once mapped, barcode and amplicon sequencing, or BAR-seq, uses very short reads. So the barcodes are only 20 nucleotides long, and you can use 50 base pair Illumina sequencing reads. And this requires very low relative sequencing power, which enables high multiplexing. And this is dependent on the machine you're using. And at the same time, this method maintains the benefits of traditional TnSeq. And so you have an antibiotic marker for selection of your library, as well as it can be relative high levels of internal replication, as seen in the multiple insertions in a single gene. So how is this method currently primarily being used? Well, it was initially developed by the Arkin and the Deutschbauer labs at UC Berkeley for high throughput functional genomics. And in their words from this recent paper, to study thousands of genes of unknown function. And as you can see from this figure, there are 32 different bacterial strands across dozens of conditions. And they're using these data to identify various phenotypes for those genes under those conditions. Some of the improvements that are currently in development for this method include the use of additional antibiotic markers and variants in donor plasmid structure to use the method in a broader distribution of bacteria. And it sounds like, again, based on Lori's seminar last week, they've also moved to fungi using an agrobacterium-mediated method. So that's actually really exciting. These massive functional genomic studies are really incredible. And they're already identifying function for hypothetical proteins and otherwise improving annotations of diverse genomes. But in our lab, we were interested in using this technique in more complex growth conditions. So for example, growth in a plant host. The conditions being tested in the previous figure were primarily various carbon and nitrogen sources, as well as antibiotic stress. And so we're interested in bacterial traits or genes that contribute to the general ability to colonize a host, as well as virulence traits that are required to cause disease. So this slide I showed previously, where essentially the infection cycle is representing the successful colonization of multiple habitats in a potential plant host. And we were interested in the diverse compatibility traits that were required at these different stages of infection. And so I'll start by talking about my main project of using RB-TnSeq to compare growth on the leaf surface, which is epiphytic growth, to colonization of the apoplast prior to disease symptoms appearing. So generally for these experiments, the first step is to construct and map a mutant population. The second is to actually test it and generate data in your conditions of interest. So all libraries require slightly different conditions depending on your organism of interest and the transposon vector that's being used. But in this case, a bar-coded transposon containing plasmid was moved from an E. coli donor library through pooled large-scale conjugations into my organism of interest, B728A. Now, the resulting population was sequenced using the modified TnSeq mapping protocol, and we were able to identify over 280,000 independent insertions within the genome, within this population. And so this works out to a median of 21 insertions per gene. From the construction of this library alone, 392 genes, about 8% of the genome, were predicted to be essential for growth in LB. And we were then able to start generating fitness data for 82% of the genes. The remaining 10% are typically small genes, or those that have too few insertions or too few reads to be able to generate fitness data. So on to the interesting part, actually testing the mutant population, generating data in diverse conditions. The methodology I used here for inoculation was either spray inoculations onto the leaf surface or vacuum infiltrations into the apoplast. And the host being used here was bean. For each experiment, I grew large inoculum cultures. These were sampled as the ×0 samples. I used these to inoculate the plants, and then harvested cells either 2 or 6 days after inoculation by sonicating the leaves or the ground leaf tissue and filtering out the leaf debris as much as possible. The barcodes from purified DNA were amplified using PCR and sequenced. This is the bar-seq step. Yeah, sorry. All right, but in planta, the challenge for these experiments was really avoiding the bottleneck effects, or the potential for the bottleneck effects. So how do we ensure that the diverse mutants in the population were represented? This is a challenge for most in vivo experimental designs. It can also be a challenge for many in vitro experiments. And so here I'm showing data from a sample experiment, which was growth in defined minimal media. This particular work was actually done here at Cornell, but I thought it'd be more interesting to show actual data rather than some of the rough estimates that we did at Berkeley. So for this experiment, I inoculated the library at different concentrations, ranging from about 100-fold coverage, so on average 100 cells per strain, ranging to 1,000-fold dilution relative to the library size. So on the left, we have essentially the relative, the number of barcodes recovered at these various dilutions. So we have 10-fold dilutions of the library, and then how many unique barcodes came out of this. The dotted line is the total library size. So as we move to the right of this dotted line, we're adding cells, but we're not adding diversity to the population. So the total number of barcodes remains constant. Moving to the left is essentially diluting out the number of cells in this population, and we're also diluting out the total diversity seen in the number of barcodes present. So from this experiment, we calculated fitness across these different samples. And so I'm showing here 2 sample genes, so genes involved in the biosynthesis of methionine, and tryptophan. These genes should be essential under these conditions. And you can see to the right of this dotted line, as we're adding more cells into these samples, we're able to calculate fitness values for these two genes. And other ones, these are just examples. But as we diluted out the population, moving to the left of this dotted line, we're no longer able to calculate, accurately calculate fitness values. We hypothesized that the range and magnitude of these fitness values is due to the different inoculum concentrations that were actually used at a constant nutrient concentration, which would potentially change the total number of generations in the experiment. Okay, so here's my photo of all the plants that I was using. So these mutant populations contain in the order of 10 to the 5th unique mutant strands. So how big do the experiments need to be? We had roughly estimated that we would ideally need at least 10 cells per strain, which works out to about 10 to the 6 cells being inoculated for a given experiment. It turns out that might actually be a slight overestimate, but we want it to be safe. And this came down to a balance between starting with enough cells to do the experiment successfully and also not overwhelming the plants with pathogen. The inoculum concentrations that I was using for these experiments were 10 to the 6 CFUs per mil for a spray inoculation and 10 to the 5 CFUs per mil for a vacuum infiltration. And so that was obviously the more limiting experiments. For each experimental replicate, I pooled 100 pots of plants. Each pot contained between 5 and 7 plants. Each plant had 2 primary leaves, which works out to about 1,000 leaves per experiment. And so this was a lot. It was doable, but it was certainly difficult. And we had estimated these values also from a successful trial experiment that was done using a much smaller number of plants at a higher inoculum density. For these large inoculations, we generated fitness data for around 4,300 genes in these conditions. So Kingsby, epiphytic growth, and apoplastic growth. The overall clustering of these experiments based on total gene fitness values looked consistent with what we had expected. For each of these plant experiments, they were repeated 3 times with 2 sequencing replicates each. Now, for most genes, disruption has no effect on overall fitness. And so here I'm showing the range of fitness values for the 3 apoplastic experiments, where each gene, the fitness value for a particular experiment is shown in gray, and the average fitness values for each gene are shown in black. Now, most fitness values are around 0. And this is a log scale, so 0 indicates that the relative abundance does not change over time. It's a log 2 scale, so a fitness value of -2 indicates that the mutant is about 25% as fit as the average strain. So from the sample distribution of gene fitness values, we're looking for the outliers. So those with the really, really large fitness effects. And as a result, we targeted primarily those highly important genes, those with a noticeable fitness defect after fewer than 8 generations. RB-TnSeq has proven to be a really powerful screening tool, especially to generate lists of genes for further analysis. So for example, I identified 33 genes specific to growth on leaf surface, 67 specific to the apoplast, and 62 genes that were highly important under both conditions. And I'll show a few heat maps like this, where the red indicates the more negative fitness values. So those are the genes that are highly important under that particular condition. And I was looking for consistency within the functional categories. And so for example, genes involved in amino acid biosynthesis, so here is an example, histidine and tryptophan biosynthesis, tended to be highly important in both plant conditions. Genes involved in the type 3 secretion system machinery were highly important in the apoplast, while individual type 3 effectors were dispensable. Polysaccharides like alginate, for example, tended to be highly important in the apoplast. Alginate has been shown to help cells tolerate desiccation stress, which supports what's some of the previous work that's shown that the apoplast is actually a relatively dry environment. Often, these polysaccharides are also important on the leaf surface, but for these experiments, the plants were actually kept at a relatively high humidity environment, and so that was not the case here. Phytotoxin synthesis and transport, such as the production of syringamycin, were also highly important in the apoplast. I thought this was a particularly interesting example because phytotoxins have been previously shown to be necessary for symptom formation, but not actually for bacterial growth. And so these data suggest that phytotoxin production is maybe necessary for at least competitive fitness in the plant. So as you might expect when you take a lab-grown population from a rich medium such as LB and compare it to growth in a complex environment, there were many anabolic genes that were highlighted in this screen. And we can actually think of these highly important genes as representing biosensor mutants. So these mutants are incapable of constructing essential building blocks, which are either provided by the plant or absent. And so using tryptophan as an example, this tryptophan auxotroph was completely unable to grow in the plant, which suggests that tryptophan is not present in this environment. But on the other hand, we found that catabolic genes were generally dispensable, suggesting that there are many substitutable carbon sources present in the plant. The most notable exception was sucrose, which is highly abundant, but even those mutants had a very moderate phenotype. This model for Pseudomonas syringae metabolism in the bean apoplast shows a sample of the variety of sugars and organic acids that can be utilized by central metabolism. But generally, we found that mutant strains lacking the ability to break down any single molecule were just as fit as the average strain. So from this study, I was able to generate genome-wide fitness data during colonization of multiple plant habitats. I've highlighted here some of these general compatibility traits for leaf colonization, ranging from sort of these general broad metabolic capabilities to the production of alginate, other polysaccharides, the type 3 secretion system, and phytotoxins. And this is a very limited list. I was interested in comparing these fitness data to previously published transcriptome data sets. Using available previously published microarray data from our lab, I could compare these expression and fitness data sets directly. The expectation was that as expression increases, either absolute or relative to a minimal medium, we would expect these genes to contribute more to fitness, as seen by the decreasing fitness values on the y-axis. But interestingly, we saw overall no global correlation between gene expression and fitness values. The genes that contributed strongly to fitness on the leaf surface and in the apoplast were generally not highly expressed or induced in planta. But conversely, the genes with the highest expression changes had fitness closer to 0. And there are certainly a few caveats for these comparisons, but I think it's an important point that's worth making when extrapolating from transcriptome data sets. So moving beyond the single pathogen system, can we scale the experimental system? So I was interested, going back to this original question of broadly, what are the strategies for success in diverse hosts? And so I tested the Transposon library in additional plant species. Tomato is a non-host, so there was insufficient growth to actually test the library, but B728A is also capable of infecting lima bean, which is in the same genus, and pepper, as well as common bean. And so within these compatible hosts are different genes required for competitive fitness and compatibility on these different plants. So to characterize the bacterial traits that contribute to colonization of these multiple host plants, I essentially repeated the previously described experiments in lima bean and in pepper. And so the fitness distributions were very similar between these three hosts, which indicates similar magnitude of growth and selection. On the right, you can see a PCA plot of the experimental conditions, which was generated using all fitness values. And the treatments are clustering here as you would predict. The question for these experiments was, are any individual genes host specific? And so we expect most genes to sort of fall in the middle. But the hypothesis was that host-specific genes would skew to either side, so either the blue or the red over here. So if a particular gene had a very negative fitness in one host, like bean, but a fitness closer to 0 in pepper, we would predict that that gene was specifically important in bean. However, we saw that there was essentially no correlation. Well, no, there was essentially no strongly host-specific genes, and there was a high correlation for genome-wide fitness between the paired plant hosts. And so maybe this shouldn't have been that surprising. There are many general plant colonization traits that would be needed in any of these plants. But at the same time, we would not detect using this method multiple genes that have maybe subtle additive contributions to fitness. With that in mind, there were genes that, while they were important in all the plants tested, significantly differed in fitness between hosts. So for example, there are certain amino acids, like including histidine and the branched amino acids, that were more important in pepper and in green bean than in lima bean. And so this suggests that these amino acids are potentially more available in lima bean. And alginate biosynthesis also came up again in the screen, where the mutants were less fit relative to the average strain in lima bean and in green bean compared to those mutants, same mutants in pepper. And so to wrap up some of this work, we're looking into whether the abundance of these amino acids in the apoplast differs, as these fitness values would suggest. So today I've talked about my work using RB-TnSeq to screen for genes that contribute to bacterial fitness during colonization of the leaf surface in common bean, as well as the apoplast of common bean, lima bean, and pepper. A major strength of this method is the ability to very cheaply generate large complementary data sets. So for example, my previous in planta work focused on a single strain of Pseudomonas syringae. Here at Cornell with the USDA, I'm working on getting this technique functional for additional bacterial plant pathogens, including the related model Pseudomonas syringae strain, TC3000, as well as the necrotrophic pathogens, Dickia didanthii and Dickia dianthicola. This gives us the power to assess not just the genes required for maximal fitness in multiple conditions, but also to screen for the common and unique functional traits in multiple bacteria across multiple habitats or plant hosts. This allows us to move beyond comparative genomics, which give us an indication of the genomic potential, to directly assessing contributions to fitness using high throughput functional genomics. And so to wrap up, I'd like to highlight really how open-ended this work is. And I'll mention that while this is simply one of many complementary tools that can be used, RB-TnSeq really has tremendous potential. So for example, measurements of gene expression compared to contributions to fitness are both really important and I think useful measurements. For my analysis, I pulled existing transcriptome data from the literature, but I think it can also be really useful to actually generate those data in parallel and actually control for some of the conditions that I was not able to control for in my analysis. TnSeq in non-wild type strains, such as deletion mutants, can be a really interesting way to better understand gene regulation, gene redundancy, and metabolic pathways. And so last year, we actually published a story that I didn't talk about today, where I constructed a transposon library in a mutant background, so one that was lacking a major drug resistance efflux transporter. And I was able to use that to identify alternative and redundant toxin tolerance genes. Sorry about that. as Paul mentioned with my introduction, the global fitness profiles can be compared to protein and metabolite measurements. And I think this is currently a very underutilized method, but probably comes down to some of the difficulties with the current tools available for actually making those protein and metabolite measurements. And then finally, if this technique or if this methodology sounds interesting to you, it's actually really easy to use if the libraries are already constructed. And so one resource, aside from talking to me, is I recommend checking out the fitness browser. So this is a website that was constructed and maintained by the Arkin and the Deutschbauer labs. And it's a little bit confusing to use, but if you click on this organisms button on the main page, it has a list of all of the strains where libraries have been constructed and published. And so you can go through, it's mostly proteobacteria at the moment. But you can go through, see if there's anything, any strains or related strains of interest. And for each strain, the website also contains all of the experiments that have been done with all of the corresponding fitness data across the genome for those experiments. So this is actually a really rich resource. All right, most of the work that I described today took place under the guidance of Steve Lindau at UC Berkeley. And I'd particularly like to highlight the work of the many undergrads that I had the pleasure of working with, and also Stratton, who was actually an early rotation student. All of the RB-TnSeq work was done in close collaboration with Adam Deutschbauer and his group. They provided the donor E. coli libraries, as well as the resources for the sequencing and the analysis pipelines at Berkeley. I'd also like to thank my current lab. I'm currently co-advised by Paul and Melanie. So thanks for the opportunity to speak today, and thanks everyone for coming. And so with that, I can take questions.
So what was the nature of the genes that increased fitness when they had insertions?
Yeah, so we actually thought there would be more. I mean, as you might expect, that property is very rare, where if you disrupt a gene, suddenly your mutant is more fit. There was the occasional regulator, so one or two component systems popped up, but those phenotypes tended to be very mild and also very limited to what conditions they appeared in. So as an example, I think one of the largest increases in fitness happened for, I don't remember exactly which one, but a two-component system on the leaf surface, but those same mutants were highly unfit in the leaf apoplast. So definitely a trade-off. Andy?
So I know I shouldn't be too much worried about cheaters.
About what?
Cheaters.
Oh, okay. Mm-hmm.
So, because I've talked about this before, but were you surprised that if you only have like 10 or 12 out of 3,000 or whatever genes involved in the type 3 secretion apparatus, that means that most of your neighbors are able to make the apparatus, and most cells are at the density that you're using, most cells are, you know, interacting? Like, were you surprised by that?
So we thought a lot about that, and the thinking currently, at least the thinking that makes me feel best about this experiment, is that when we did the inoculations, we did expect them to be in relatively high density, and that you would expect more than one single cell to be interacting with a plant cell, just due to the scale. And so if that were the case, then you should get that complementation between those mutants that cannot deliver effectors and those that can. And so we were pleasantly surprised, essentially, to see that those secretion system mutants were coming up in our screen. And what my interpretation of that is that the inoculum density actually was low enough that they didn't get complementation at the beginning of the experiment, which was when most of the growth was taking place. You know, growth curves in the apoplast, you typically get a lot of growth pretty quick, and then it plateaus a little bit. And so my expectation is that there probably is complementation later on in the experiments, but all of the growth is happening at the beginning, and that's actually what we're measuring. And so that's without actually looking to see, you know, that's sort of where our thinking was.
You should ask if there are questions.
I don't know how to do that.
Or just ask them the microscopist and they'll use themselves.
What
Are there any questions from Geneva? Okay.
are we looking for?
No, we just asked them and they tell us.
Okay.
It's low tech. So I had a question related to the cheater question. Could you go back to the slide with the algae and that the toxin?
This one?
Yes. So I think it's kind of interesting that the level of fitness for the individual genes within the two, we'll call them modules, is somewhat varied. I imagine that could be statistics. But I noticed, for instance, that algae is loss of algae is very detrimental for fitness, and we know that algae is also a major regulator of other systems.
Mm-hmm.
And so you see these individual genes that contribute more to fitness than others, and I'm wondering, so there's cheaters, there's other functions for some of these genes potentially. Do you see any patterns? Is there any way of sussing these things out?
Like looking at regulators specifically?
Well, I don't think that all of these genes that are contributing to fitness are regulators necessarily.
Oh,
so are there other,
no. Yeah.
I mean, are there other secondary functions that some of these things might be playing that you can somehow mine this data to figure that out?
I guess I realize I have not been repeating the question, which I should have been. But in this case, to see if I actually understand the question, you're asking if these genes have other roles in addition to their like enzymatic functions, or like how you would screen maybe for additional function.
I'm not putting you on the spot and asking you about these particular pathways. I'm just noticing that, for instance, algae, which we know that has, and so have you run into other instances, or are there patterns that we can be looking for in the data that might give us hypotheses for further experiments to test? You know, these things might be important in other ways.
Yeah, so I'm not sure if from these data you can get a sense of if these genes would be functional in other ways. I mean, one of the big caveats for the analysis is that, especially in planta, the fitness values can be highly variable due to the nature of the experiments. And so we talked a lot about, Steve and I talked a lot about trying to pull out genes that had more moderate phenotypes. Because in this case, most of the analysis I did was pulling genes that had these really screaming, huge fitness defects, because that's essentially the low-hanging fruit for these analyses. If anything, that's one of the challenges with these experiments, is that the analysis that I've done is basically scratching the surface. There's a lot of other stuff that can be done, working within the issue that these results can be really variable. But in terms of regulators, at least, I think that is where you can get some really interesting stuff, looking at what happens downstream when you start disrupting these pathways. Although that can be a lot. Yeah, we can talk about that more, because I think maybe I'm not totally understanding.
No, I think you do understand the question, but there are some ideas for mining the data. Okay, so before we thank one more question?
One more question. Sorry.
Yes.
So for your spring inoculation data, do you find the chemotaxis genes to be strong outliers, having very negative impact on the fitness?
Oh, that's a really good question. I think motility was an interesting example for the leaf surface because what I was seeing did not fully line up with what people have seen in the past.
Yeah.
I think the reason for that might have been, so for what I saw was primarily the question regarding chemotaxis and motility on the leaf surface. I saw generally flagellar mutants tended to be more fit. Those genes, those proteins use up a lot of energy. And the assumption that we were making based on the fitness values I was looking at was that the inoculum density was high enough that cells didn't need to move around that much. If they landed in spots on the leaf surface that had nutrients, they grew. And if not, they were probably immediately outcompeted by other cells that did. And so from these experiments, I didn't really see any chemotaxis genes popping up as being important. But I think that might have been more of an experimental artifact than anything related to the importance of those genes on the leaf surface, which have been shown previously to be really important.
Sorry.
One more question?
So, Tyler, so the inoculations were community inoculations, correct? You're not inoculating individual strains.
So the inoculations were of a pooled mutant population inoculated simultaneously.
So then, and this is going back to cheaters also, but, and so in order to verify that you've got these, actually the fitness defects are not necessarily due to competitive interactions, but actually due to some type of inhibition in the pathogen's ability to infect. In order to separate those two, you would need to run these experiment using the bacteria mutants solo, right? To avoid competitive interactions.
Yeah, so this method is a really good screening tool to generate a lot of data at the same time. But you're right, in order to get a better sense of the phenotypes of any single mutant, it is really valuable to make a targeted deletion, test it by itself so that you don't get these community effects. And I certainly did that for some of the mutants that I was looking at. Again, mostly those that had really large fitness defects. But that essentially became the limiting step, is picking out genes to actually validate. Yeah.
So it's worth noting that the software that's used to do the analysis and statistics on these results, we have it working at this point in our lab. So if this is something that you guys are, if anyone is interested in looking at this, please talk to Tyler. All right, let's thank our speaker.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.